Okay, good afternoon, students. And today we will rediscuss the chapter Microbes in Human Welfare. Actually, students, the term 2 syllabus we have completed, and now from the beginning, okay, we will do the rediscussion, or we can say revision of the chapter Microbes in Human Welfare. Then again I will proceed to biotechnological principles and processes. After that again biotechnological applications. Okay. So today we will start with the rediscussion or the revision of chapter 10, that is Microbes in Human Welfare. Okay. For this, let me open the slide of the chapter. Okay. Within a second, the slide will be shared with all of us. Okay, yes, it is there on the screen. My screen is visible to all. My screen is visible to all. Slide is visible. Okay, fine. Okay. Okay, I got your remark. Now, students, microbes in human welfare. To begin with the chapter, we will start with a preliminary idea. I think all of you will be able to answer my question that what microbe is. What is microbe? What is microbe? Yes, through your chat box you give the reply. How we can define a microbe? This is very simple question for class 12 students. Organism that can't be seen by naked eye. Okay, Prashasti. Supriya, we should say definitely we should not confine our answer with small or big animal which can't be seen by naked eye. Okay, there are small animals or small organisms also which are macroscopic. That means which can be seen by the naked eye. Okay, which are visible by microscope. Okay, fine. So this basic preliminary knowledge you all are having, that this is a microbe, that is these are the, we can say, the organisms which can't be seen by the naked eye. Then what, or we can say which group of organism, or there is we should say there is a diversity among the microorganism too. Why we are saying there is diversity among the microorganism? Why we are using this word? Why we are using this word? There is diversity among microorganisms. Why we are using this word, that there is diversity among microorganisms? Why we are using this word, there is diversity among microorganisms? Please give your reply through chat box. Anyone? Okay, Supriya. Very good. Protozoa, bacteria, fungi, plants. Okay. Okay. Yes. Okay, fine. Okay. Yes, because the number of the organisms are included under this group. The number of organisms are included under this group. Okay, fine. Yes. Number of organisms like bacteria, protozoa, fungi, microscopic plants, virus, viroid, prions. So these are the microbes which are present in the living world, and in this chapter our focus will be how microbes are helping humans, or in which way the microbe is beneficial to us, either in our household product or as an industrial product or in the sewage treatment or in the biogas production. So now we will come to our next slide. Presence of microorganism. Where do you find microorganisms? Is there any particular place where you will be finding the microorganism? Or if I ask you, give a single word for the presence of microorganism, what you will say? Everywhere. Very good, Divya, Shupriya. Very good. They are everywhere. Okay. Yes, they are everywhere. That is omnipresent. Omnipresent. They can even survive at a high temperature, deep in the soil, under snow. Different groups can be present. Okay. So now we will be coming to our next section, that is the importance of, or we can say the microbes' importance in household products. Students, this part, all of us, we know that a very common use or a very simple use of microorganism in the household product is curd. Very good, Anshika. Before the question only you have answered. Curd. Which bacteria is present in the curd? Yes, correct, Ankit. Milk to curd. Which bacteria is present in curd? Which bacteria is present in curd? Okay, lactobacillus. Okay, lactobacillus is present in the curd. Now, for making the curd, what we do? We take lukewarm milk and we add small amount of lactobacillus to it, which act as a starter. Okay, that is, this lactobacillus or the lactic acid bacteria, it acts as an inoculum. Correct, Anshika? So they will convert milk into the curd. And during the growth, what is produced? This lab, that is the lactic acid bacteria, produces acid that coagulates and partially digests the milk protein. Not only that, there are some benefits of the lab. First, it increases the vitamin, or we can say increases the content of vitamin B12 also, and the second thing is that it checks disease causing microbes in the stomach too. One very important thing, students, in this image which I have shared with you all, here you can see lactic acid bacillus in the form of tablets. Students, see, long-term use of the antibiotics. Okay, what are antibiotics, students? Before going into this topic, let me let you answer what are antibiotics. How we can define antibiotics? How can we define antibiotics? Give me the proper definition of antibiotics in the chat box. How can we define antibiotics? How can we define antibiotics? No, Sneha, I want the definition. Okay, you are saying in the functional aspect, fine. This will be also considered, but tell me, how can you define antibiotics? Very good. Correct, Divya, correct, Shupriya, correct, Ankit, correct, Anshika, very good. Correct, Minal. Okay, these are the, yes, the chemical substances, definitely the chemical substances which are being used in the body to produce antibody to check the growth of the microbe inside the body. Now, student, you must have or you should know that whenever you take antibiotic for a long time, then your stomach upset or either the dysentery, this problem arises. Have you noticed this? That a long-term use of the antibiotics have some side effects on our digestive mechanism. Do you agree or not? Do you agree or not? This reflects your observation. Okay. So, why this happens? Can anyone say why long-term consumption or intake of antibiotics hampers our digestive mechanism? Can anyone guess? Can anyone guess why long-term intake of antibiotics disturbs our digestive mechanism? Very good, Supriya. Correct answer. Prashasti, we have to be more specific. Okay, Anshika. Actually, student, in the gut of our body, or we can say in the intestine, beneficial microbes are also present. That is lactic acid bacillus. Now, this lab or the lactic acid bacillus is important in the digestive mechanism. Now, when we take antibiotics in the gut, these beneficial microbes is killed, and that is why sometimes when you suffer from dysentery or the loose motion, you must, if you notice, whenever the medicines are given to you, if you notice that sometimes if you will see the composition of the medicine, it is written lactic acid bacillus are there. So what these tablets or what this medicine contain? They contain lactic acid bacteria, and when you consume it again in your body, in the gut, this lactic acid bacillus bacteria or the beneficial bacteria is again harbored, or it is again being present over there to check the body digestive function proper or smooth conduction. Is it clear to all? Is it clear to all? Yes. So you can see in the last point that in our stomach too, the lab is very beneficial in checking disease causing microbes. So we can say, if remember student, this question is very important also, that what are the two benefits of lab? So one, it increases the vitamin B12 content. Second, in the gut, it checks the growth of the disease causing microbes. Okay, so now we are coming to the next household product, or we can say the microbe used in the next household product. The next product is Dove. Okay, okay, yes, Dove. Let me, where you have observed Dove, this is one of the products which you and we are eating in our day-to-day life. So where do you find this product? Where do you find this product? Where do you find this product? Ankit, you have entered the class with two profiles. Ankit, please respond. You have entered the class with two profiles. Okay. Please respond in the chat box, Ankit. Is there any problem with your device? Though I admitted you, but if okay, net issue. That's why I asked. Okay, fine. Okay, the Dove. Yes. Where do you find this product? Where do you find this product? Okay, bread. Okay. Yes. Dough which is used for making bread is fermented by using baker's yeast, and that is Saccharomyces cerevisiae. So definitely the dough is formed by fermentation, and the CO2 released during the process of the fermentation gives the puffy appearance to dough, and that is why it is used in the production of the foods like idli, dosa, the bread, because of this puffy appearance. So a question also comes on this, that how the puffy appearance appear in dough. So the puffy appearance or the puffy appearance is due to the CO2 produced during the process of fermentation. Okay. Now the next household product. The next household product is, just a minute, let me change the page. The next household product in which microbes is helping is toddy, which is a traditional drink in the southern India, which is made by fermenting sap from palm. What is sap, students? This from your class 10. We are studying, not class 10, from 9. We are studying. What is sap? You must have heard about the mechanism in plants, ascent of sap. Good, Supriya, very good. Quick answer. Yes, quick answer. Water and minerals. Very good. Water and minerals are togetherly called sap. Very good. So, water and the minerals, correct. By fermenting the sap from palm, toddy is prepared. After this, the next household product which we are going to discuss is cheese. Okay. The next household product which we are going to discuss is cheese, which is formed by partial degradation of the milk, and the Swiss cheese is produced due to the production of the large amount of the CO2 by a bacterium called Propionibacterium shermani. Be very careful with the spelling, students. Propionibacterium shermani, this is a long spelling, and your chances to confuse with it is more. So this is the one. Now this cheese, and one more cheese is there, that is the Roquefort cheese, which is ripened by growing a specific fungus, and by growing a specific fungus for a particular flavor. During the classroom also I told you what is the name of this fungus. Can you recall, student, what is the name of the fungus? What is the name of the fungus? Penicillium roqueforti. Yes, correct, Supriya. Very good. Students, parallelly, you keep your previous notes also side by side with the revision class, so that you can easily correlate with it. Okay. So, total, can you sum up what household product we have studied? Togetherly, we have studied 4 household products. Can you recall? Can you recall the 4 household products? The 4 household products which are being used: dove, curd, toddy, and cheese. These are the 4 household products which the microbes are helping in the production. Now, on the basis, or we can say on the basis of the 4 household products which we have discussed, now we will be discussing one. I am showing you one flow chart which is based on the microbes used in the household product, and let us first do this worksheet, and then again I will come back to this slide. Let me show the worksheet. Yes. Is my worksheet visible to all? Now, student, this is a flow chart, okay? This is a flow chart. The first step is milk is incubated with curd. The second step is lab grow in milk, after which it's labeled A. That means you have to identify A. After A is the step coagulation and digestion of milk protein. Then you have further step improve nutritional quality by B. Improved nutritional quality by B. And what does A and B signify in the flow chart? Expand the word lab. Milk starts to coagulate when lab is added to warm milk as a starter. Mention any other two benefits that lab provides. So all of you first write down this flow chart. All the students write this flow chart. Write down this flow chart. The first step is milk is incubated with curd. Next step, lab grows in milk. Then third step, the keyword you will fill A, and here one clue I can give you, there is a production word. After which, fourth step, coagulation and digestion of milk protein. Fifth step is improved nutritional quality by B. Improved nutritional quality by B. The first question, what does A and B signify in the flowchart? What does A and B signify in the flowchart? Student, parallelly you give me the response that what does first A stands for and what does B stands for. In the chat box you send me the reply. In the chat box you send me the reply. Okay. A is, I want, see, near about, see, 10 students are present in the class. I want the reply from each and every student. I got the reply. I am just seeing the chat box. Remember, don't answer all the questions togetherly. One by one answer, and I want the reply from all the 10 students individually for each question. Yes, one reply Supriya has given. Okay. After some time I will declare the answer. Yes, Supriya, your answer is correct. Your answer is correct, Supriya. I am waiting for further responses. Okay, Minal, I repeat the question. I repeat the question, Minal. See, this flowchart is how curd is formed. Okay, the first step is our milk is incubated with curd. Milk is incubated with curd. This is the first step. The second step is lab grows in milk. Third step we have to answer, which is A in the figure. Next step is coagulation and digestion of milk protein. And after that, improved nutritional quality by B. So, what does A and B signify in the flowchart? And you have to expand the word lab. So, first, let us focus with the first question. What does A and B signify in the flowchart? Okay, Divya? Divya, you didn't answer for the first one. Yes, Supriya, I got your answer. Supriya, I got your answer. I am asking to all the students regarding question 1. Wait, let me check the responses. Yes, Ankit, no need to remind me. I have seen. I have seen. Yes. Okay. Yes. Okay. Correct. A is correct. Yes. Correct. Yes. A is lactic acid bacteria lab and B is increase in vitamin B12. Correct. Now expand the word lab. Next, expand the word lab. Expand the word lab. Lactic acid bacteria. Lactic acid bacteria. Correct. Very good. And the third one, milk starts to coagulate when lab is added to warm milk. When lab is added to warm milk as a starter, mention any other two benefits that lab provides. Mention any other two benefits that lab provides. Mention any other two benefit that lab provides. Very good, Ankit. Very quick. Very good. Checks the growth of the microbes in the gut and also increases vitamin B12. Very good. Okay. So this type of flowchart question also, this is very important as because in the board examination maximum time this type of questions are asked. Okay. Now let us move back to our, correct Divya? Correct? Okay, very good. Today, Raj Nandini, I am not getting any response from your side. Raj Nandini, you have not responded today. Is the class properly audible to you? Raj Nandini, please respond. Okay, fine. Okay. Okay, now let us come to our previous part. Now let us come to microbes in industrial product. First, the beverage industry. Okay, first the beverage industry. Now, production of the industrial scale requires growing microbes in very large vessels called fermenters. And one of the products which is the fermented beverages, so this product we are using for, or we can say we are using which organism? The yeast. So definitely student, this as a household product also we have given an example. These are for the household product also we have given an example because we are using that product in the house, but it is prepared in industries. So the yeast Saccharomyces cerevisiae is used for making and commonly called brewers, used for fermenting malted cereals and fruit juices to produce ethanol. So when it produces ethanol, student, what type of fermentation it is called? When it is producing ethanol, what type of fermentation it is called? What type of fermentation it is called? Because we are knowing in the production of the wines, they are used. Yes, okay, correct. Wine and beers are produced without distillation, whereas whiskey, brandy, and rums are produced by distillation of the fermented broth. Then we are coming to the next group of chemicals, or we can say the next group in which the microbes are helping. And the first very important one is the antibiotics, which in the beginning you have defined. The antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. Penicillin, first antibiotic to be discovered, and it was a chance discovery. Alexander Fleming discovered this while he was working on Staphylococcus bacteria. Once observed a mold growing in one of his unwashed culture plates around which Staphylococcus could not grow. So, why did it the Staphylococcus was not able to grow, students? Can you guess? This was Alexander Fleming's observation. That is in our NCERT textbook also. It is called as a discovery of penicillin. So while Alexander Fleming was working with the bacteria Staphylococcus, he observed a mold growing in one of his unwashed culture plates. Okay, in one of his unwashed culture plates, the bread mold, or we can say the mold, that is a fungal. Here we will be saying it as mold only because it is a specific that was growing in one of his unwashed culture plates. Correct, Divya? Correct, Supriya? This was due to the growth of the penicillin. Staphylococcus was not able to grow there. Correct, correct. Yes, and it named it as penicillin after the mold Penicillium notatum. And later, Ernest Chain and Howard Florey made its full potential effective antibiotic. And due to which these three scientists, Fleming, Chain and Florey, they even got the Nobel Prize in 1945 for their discovery. Okay, and the diseases which can be cured by the antibiotics includes nowadays a wide range of the diseases like plague, whooping cough, diphtheria, and even leprosy. Okay, so this was about the wonder drug penicillin. So in case of the antibiotic section, you have to remember the definition, the penicillin, the discovery of the penicillin. Okay, is it clear to all? Yes. Now here, students, uh, this is very important part, as because during our offline class, when we were doing this chapter, some part of this chapter might have got erased from your memory, as because this chapter we did during the time when term 1 examination didn't happen, because of the fact that this chapter was not included in term 1, though that time we have completed. If you can remember, and if you see the class notes, I have made you write here three tables. And in the first table, you all have written organic acids and alcohol produced by microbial metabolic actions. Second table was common important enzymes produced, and other was a bioactive molecules. Now, when we are discussing the household products, when we are discussing the microbes in the industrial product, we have to consider this also into account. Okay, these are the chemicals, the enzymes, and the other bioactive molecules. Okay, so the first one, what is the name of the organic acid? Citric acid. Which microbe, which microbe is producing this acid? Aspergillus niger. And one more column which we have included within this is which or what is the group of the microbe. So Aspergillus niger is a fungus. The second, acetic acid. What is the common name of acetic acid, students? We popularly know acetic acid as, can anyone say? Very good, Sneha, very quick answer, very good. Rajnandini, very good. Correct, Divya. Correct, Ankit. No, 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 Ankit. Think it's vinegar. Okay, vinegar. We are using in the preparation. We are using in the many of the household food materials. Yes, yes, Ankit, like that. Now, the next one is, so acetic acid, the vinegar, is produced by the bacterium Acetobacter aceti, and this is a class of the bacteria. Now, the Clostridium, the next is butyric acid. Butyric acid is produced by the bacteria Clostridium butylicum. Then comes your lactic acid, which is very common one. It is produced by the bacteria Lactobacillus. And the last one, that is the organic acid ethanol, is produced by the Brevus is Saccharomyces cerevisiae, and this is the yeast. The yeast belongs to which group? Yeast belongs to, I should say, which kingdom? Yeast belongs to which kingdom? Correct, Supriya, very quick. Correct, Divya. Correct. Okay. Okay, very good. Very good, Rajnandini. Yes, it is a fungi. Correct. It is a fungi. Correct. So here in this table, we have studied the chemicals. Three tables, actually it is a three tables, chemical, enzymes and bioactive molecule. One by one we will do this. So the chemicals we have done, togetherly we have studied 5 chemicals: citric acid, acetic acid, butyric acid, lactic and ethanol. The different microbes: citric acid produced by Aspergillus, acetic acid produced by Acetobacter, butyric acid produced by Clostridium, lactic acid produced by Lactobacillus, and ethanol produced by Saccharomyces. Okay, so students, today one assignment already, which this question regarding the lab I have provided, you will complete that one, and you will also prepare this very table which contain the name of the organic acid and the ethanol, alcohol. And next class we will be continuing with the other 2 tables of the enzymes produced by the microorganism and its importance, and the bioactive molecules. Okay. Okay, students, thank you all for today's class.